All right. The ear. I had given some anatomy. I don't remember how much of this I had covered before. I think I had left it at the petrous temporal bone. We talked about the bony labyrinth and the two portions of the inner ear, the cochlea that gives rise to hearing, and the vestibule that get the vestibular system or complex that gives rise to our sense of acceleration or motion and orientation, which are all important to giving us our sense of balance. All right, so first, the organization of today's lecture is going to be, I'm going to go through hearing first. I'm going to go through the specific process of hearing and down to the cellular components of it, cellular correlates. Then I'll move on to the vestibular system, and I'll get as far through that as I can in the time that I have. Each of these will conclude with the projection pathways through the brain that get us to the cortex. All right, so first, the cochlea. Let's take a closer look at the cochlea. So what an amazing thing. Look at that thing. It looks like some super elegant spaceship or a hobbit house, solar house of the future or something, living in our heads. Taking a sort of abstract view of the inner ear, really it's just a chamber that is labyrinthine. It's a single chamber that has all these labyrinthine paths that coil and twist this way and that. The cochlea itself is really just a giant spiral, named the cochlea, as you may be able to infer, because it looks like a conch shell. It's shaped very analogously to a conch shell. However, there is a difference. A conch shell has a single spiral that curves up towards its apex, and the cochlea actually has three, really. There are two bony passages and one membranous passage. The first bony passage is called the tympanic duct, and it spirals around. In this picture, the bottom, to say inferior and superior is not really meaningful here, but it spirals around to the apex and then joins at the top with the vestibular duct. And the vestibular duct spirals around downward towards the base of the cochlea. So if you were a teeny tiny little person, you could stand on this and walk all the way around and then get to the top, and it would spiral back down and you'd come to the other. You could begin at the round window in the middle ear and continue through the tympanic duct all the way down to the oval window at the vestibular duct. In between these, wedged in between these, is this membranous chamber, and we call this the cochlear duct. The cochlear duct is where the actual hearing organ is, and we'll take a look at this in more detail in a moment. But the specific organ is called the organ of Corti, the organ of Corti, and it's embedded in this membranous duct. Now, all of these ducts are filled with fluid. They're all fluid-filled. The fluid that is inside the bony labyrinth is called perilymph. Perilymph. P-E-R, here's the word. Perilymph. P-E-R-I-L-Y-M-P-H. And then the membranous labyrinth is filled with endolymph. Endolymph. The organ of Corti, I said, is where the sensory receptors reside, and we'll see how that works in more detail in a moment. But that sensory information that is gleaned at the organ of Corti then passes to the spiral ganglion. So here are these spiral ganglion cells. You can see them. You can see one here, maybe some here. The spiral ganglia is where the cell bodies of the hair cells, the inner and outer hair cells, which are the sensory receptors of the cochlea, reside. And then they project fibers along the vestibulocochlear nerve, specifically the cochlear branch of the vestibulocochlear nerve. They're really two nerves, but we call them the same, we lump them into the same cranial nerve because they blend into one another as soon as they emerge from the organ that they're innervating. I think that is, well, I guess I could say this on this diagram. The membrane that separates the tympanic duct from the cochlear duct is called the basilar membrane, And then the one that separates the vestibular duct from the cochlear duct is called the vestibular duct. There is a third membrane in there called the tectorial membrane. It doesn't separate anything. It's part of the organ of Corti, as we'll see in a moment, and it comes into contact with the hair cells. So we'll see that in the next slide, I think, here. Yeah. So taking a closer look at one of a simple cross-section of a single turn of the cochlea, we can see the vestibular duct on top, the tympanic duct on the bottom, the two bony parts of the bony labyrinth, and then the cochlear duct in between, wedged in between, which is part of the membranous labyrinth. And then there is, in the inset here that's pulled out, we have the organ of Corti. Corti is an eponym that refers to the anatomist that first described this structure. So if we look at it, they're emerging from the basilar membrane that separates the tympanic duct from the cochlear duct. There is a layer of epithelial cells, it's a simple columnar, a set of simple columnar epithelial cells that has embedded in them these specialized cells, sensory receptor cells. There is a row of inner hair cells and then 3 rows of outer hair cells. These cells, we'll take a closer look at what a hair cell is in a moment, but they spiral around. There's just a long chain of them that spirals around the whole length of the cochlea. And then hanging right above them is this tectorial membrane. The tectorial membrane is sort of protects the hair cells and aids in their stimulation, and specifically helps fine-tune the fluid dynamics that occur in the cochlea when there is a vibration. Yeah. Yeah, they call them hair cells. It is not actual hair. So hair, an actual hair of any sort, even a fine hair that maybe, a fine hair on your cheek or something, would be far larger than that, right? So a single hair is surrounded by many, many epithelial cells, and it's a large thing. They call them hair cells, and we'll see in a moment why they are called that, but it's not actual hair. They're projections, these stereocilia that emerge from the cell. They are specialized projections, kind of like villi or something, that microvilli that project from the apical surface of these epithelial cells that have become adapted to perform this function. All right. Any other questions? Good question. All right. So here is a scanning electron micrograph of these cochlear hair cells. And you can see that they do have slightly different structures. There's this row of inner hair cells, and then these arrays of outer hair cells. The specific patterning that you see is a consequence of physics that's involved here, the fluid dynamics that are required for picking up vibrations that happen in the cochlea. We'll see how this happens in a bit, but just wanted you to see what a pair of happy, healthy cochlear hair cells looks like. Okay, so this is how it actually works. Some sort of sound happens. We have a tuning fork here, for example. And what is sound? But it is, in fact, just pressure waves that are being caused in the atmosphere around you, in the air. So, like the sort of cliche, no one hears you scream in outer space, or whatever the name of a cheesy horror, space horror novel or something, this would be true, because there would be no pressure waves without the presence of air. So I guess like all those Star Wars movies where they're shooting laser beams and you can hear it all going on out there is actually kind of untrue, unrealistic. All right, but the pressure waves, they have a certain frequency to them, right? And what is frequency? Frequency is just how many waves per second. The unit of per second is hertz. A hertz is per second. The inverse of which is simply wavelength. It's the difference between peak to peak or trough to trough, whatever. Depending upon what we perceive of as pitch, so a high-pitched sound versus a low-pitched sound, those correspond to the length of a wavelength. So high-pitched sounds are short wavelength, low-pitched sounds are long wavelength. Whereas the amplitude of the wave is what corresponds to our perception of volume, the intensity of the sound. So you can have pressure waves that are being made in the air that are not very intense, so not a very loud sound, or you can have pressure waves at the same frequency that are really intense. So we're moving much more air. Whatever is putting off the sound is moving much more air with each vibration, and that would be perceived as a much louder sound. So these pressure waves enter, the auricle here, your external ear, the auricle captures those pressure waves and channels them into the external acoustic meatus, or your ear hole, where these pressure waves impinge upon the tympanic membrane, which is just like the membrane of a drum, and it vibrates sympathetically with the pressure waves. All right. This vibration is transduced from the tympanic membrane by the auditory ossicles, the malleus, the incus, and the stapes. Malleus meaning mallet or hammer, incus meaning anvil, it's the word for anvil, and then stapes is the word, the Latin version of stirrup. So the stapes transmits that vibration into the oval window. You'll remember that the oval window is the beginning of the vestibular duct, and the round window is the end of the tympanic duct. In between them is this cochlear duct, this membranous labyrinth. Well, the vibration of this stapes inserted into the hole of the oval window causes the fluid in that vestibular duct to oscillate at a certain frequency, right? There's the frequency of vibration that corresponds to the movement of these sound waves. Well, because of the structure of the cochlea, whatever that frequency is, it's going to vibrate a part of the membrane, in the basilar membrane of the cochlear duct, at some point along this spiral. So if this vibrating right here at this point in the cochlea, that corresponds to a specific frequency of sound. Maybe a higher sound would be over here. Maybe a lower sound would be over here. Actually, I forget what the mapping is. So then the hair cells that are embedded in that part of the basilar membrane in the organ of Corti get stimulated, and there is a labeled line code where that signal is sent via the cochlear nerve to the brain. So here is a little bit of a diagram that helps you to conceive of that. In the top picture is the ear at rest. And so it looks like, I was correct, that high frequency sounds, short wavelength, high-pitched noises, high frequency sounds are, they have shorter wavelengths, and because it has shorter wavelength, it travels not as far into the cochlea. Longer wavelengths set up standing waves further into the structure of the cochlea. So the vibration first pushes in, and it's going to distort the basilar membrane away from the inward push of the round of the oval window and the stapes. And in so doing, it's going to cause the round window to go in the opposite direction. So fluids are incompressible, right? You can't; they're not really compressible. And because of that, if you're decreasing or if you're pushing in here, it's going to bulge out this way, and something's going to have to give. So for every displacement this way in the oval window, there's going to be a displacement in the opposite direction on the round window. And then vice versa, back the other way. So this vibration gets set up here, stimulating those outer hair cells and inner hair cells of the organ of Corti, which gives us the sense of hearing. How does this actually happen? Well, all of it is mediated by mechanically gated potassium channels. So in these quote-unquote hairs that Omalara was questioning, they are actually just projections, cellular projections from the apical surface of these hair cells that at the very tips of them have these potassium channels embedded in them. And a potassium channel, the gate of the potassium channel has this linker protein that connects to the next stereocilia up the chain. Have I said the word stereocilia yet? So stereocilia is the actual name for these hairs. Stereocilia. So stereo meaning that they have they're cilia that have a spatial orientation about them. If a stereocilia bends one direction, you're going to have a different response than if it bends in the opposite direction. So if these hair cells bend in this direction, this linker chain, because we're changing the geometry of the one stereocilia with respect to its neighbor, it's going to rip open the door to this potassium channel and allow the influx of potassium. When that happens, that's going to cause the depolarization of that cell and lead to stimulus. So here is the cortical projection pathway. The sounds come in of whatever complex frequency range, whatever pattern of sound is coming in. They trigger the nerves of the cochlear branch of the vestibulocochlear nerve, and they pass down through what is called the internal auditory meatus. It's a part of the petrous temporal bone, and have their first synapse in what's called the cochlear nucleus of the medulla oblongata. From this point, we have fibers that ascend to the inferior colliculus of the mesencephalon. So we didn't really get a chance to talk about neuroanatomy that much, but this is the pineal gland, and this is the superior colliculus. I have talked about the superior colliculus. Do you guys remember when I talked about the superior colliculus? It was actually just the last class. The superior colliculus receives visual information. And then there is the inferior colliculus, which receives auditory information. The significance, I'm just pointing this out because it's kind of interesting. The pineal gland is where you get, it makes melatonin and helps regulate the circadian cycle, your wake-sleep cycle. And its nearest neighbor is the superior colliculus, which is part of this little quartet here. It's called the corpora quadrigemina as a group. But the superior colliculus, it means that visual stimulation that you have affects your sleep-wake cycle, which should make sense. Should make sense to you. But anyways, the inferior colliculus is right below it. And then there is a projection out of the inferior colliculus to what's called the medial geniculate nucleus. Geniculate to genuflect. What does it mean to genuflect? You guys know that word? What does that mean? Neil, who said it? Thank you. Yeah, so the medial geniculate nucleus is the knee-shaped bump on the side of the thalamus. We have a synapse there, and our third-order neuron then projects from the thalamus up to the auditory cortex. The auditory cortex is just a somatotopic map, although it's not somatotopic, meaning the body. It's an audiotopic map. It's a map of all of the sound that we are able to hear that is laid on top of the temporal lobe. So that's the little thumb in my little map of the brain here. It's right here along the top of that temporal lobe. All right, so that's the auditory projection pathway. For us to be able to sense that we heard something, there needs to be that synapse in the medial geniculate nucleus. From the inferior colliculus, there's a lot of other projections out. That's kind of a clearinghouse for auditory signals. So it's going to communicate with the cerebellum, and other innate motor functions. Say you hear a bang out of behind you, rather than thinking, "Oh, that was a bang, I better move myself." There's like an automatic response. Your body immediately responds to that as a reflex. So that is a non-conscious reaction. You are conscious of it, but you are not choosing to respond. It's a reflexive action. Okay, so the ears, I've waxed poetic about the eye for 2 lectures, but let me just give the ears their due. There is no other organ in the body which has the same dynamic range as the ear. So this is what's called the Decibel Scale. This is how we measure the intensity of stimulus that the ears are able to pick up. It is a logarithmic scale. It's a logarithmic scale. So, for example, 20 decibels. So 10 decibels is 10 times the power of no decibels. 0. So 0 is not no vibration at all. 0 on the decibel scale is described as the lowest audible sound, the quietest possible sound that you can hear that you say, oh, I hear a sound. 10 times that is 10 decibels. 100 times that is 20 decibels. So, for example, sitting through one of my lectures is probably something like 50 or 60 decibels. So I, right now, am speaking at you at about 1 million times more intensity of sensated, sensory input than it would be at your quietest possible audible sound, all right? Even that, just 1 million times more intense, if the lowest level of light that you could perceive, if you went 1 million times more intense in terms of the light, you would be literally burning the backs of your eyeballs out. And that's nowhere near the loudest sound. Luckily, you could listen to me all day without receiving much damage to your ears. However, if you we had the door open and we heard Whitney as well, so it was more like a busy restaurant, it could be a little bit dangerous. There might be some damage if it was nonstop, continuous assault on your ears. More than 8 hours hanging out in the subway, you can expect some damage. So then, noisy home, if you got a couple kids at your house, it's fun, but you can do some damage to your ears. Lawnmowers, et cetera, et cetera. Chainsaw, more than 2 hours. I have a friend who's an arborist. He's that guy that lives in a log cabin next to exit 127 out there, and you have to shout at him to have a conversation. For all of you into heavy metal, you may be in immediate danger, which sounds like a great name for a heavy metal band. Or if you're into sandblasting, also kind of a dangerous thing. So, gunshots. I don't know. I was watching. So there's this comedy show, the comedy cartoon Archer. I'm not suggesting you watch it. But one of the running gags on there is that the guy's got tinnitus because there's constantly gunshots going off around him. And if you unfortunately find yourself on a rocket launching pad, hearing loss is certainly inevitable. Please be careful around rockets. But the point here is that, dear Lord, what is that? So this is million, billion, trillion, quadrillion. That's 10 quadrillion times the sensory intensity from the quietest sound to the most potent sound that you can hear before your ears start to bleed. It's pretty incredible. I think it is. So, just the entreaty to your better judgment. Try to have healthy hair cells. This guy looks cool down there, rocking out in front of his Marshall stack, but his I don't know, maybe the name of his band is Noise Damaged Hair Cells, because that's what it can look like. I'm guilty of damaging my hair cells a little bit myself. All right. Are there questions on hearing? I've got about 10 minutes to tell you about balance. All right. So this bony labyrinth extends from the cochlea into the vestibular system, as does this membranous labyrinth. So indeed, the same paradigm holds in the vestibule. We have the semicircular canal. The vestibule semicircular canals. If we take a transect of a semicircular canal, you can see this bony labyrinth filled with perilymph, and then there's going to be a little membrane that's suspended in here, which itself is filled with fluid, the endolymph. The vestibule itself is what gives us our sense of gravity and acceleration. So there's all of this is by variation on a similar theme as to what I described in the cochlea. There are these hair cells that are going to have stereocilia on them that are going to get physically stimulated. So let's take a closer look at these hair cells. They're the basic receptor of the inner ear. We saw the stereocilia in the cochlea. There's a little bit different arrangement here in the vestibular system. So they're going to give us information about what direction, spatial direction, we are moving, and it's going to give us information about the intensity or strength of this mechanical stimulation. So this right here, what you see is a cluster of hair cells, of course, in a colorized image that you might find in in the vestibular system somewhere. So let's start with the Crista Ocularis. The Crista Ocularis is the functional organ at the base of the three semicircular canals. Now, I don't have a model of this, and it's definitely probably appreciated better in the anatomy lab with a model in your hand. But sometime when you do get somewhere where you're able to have a little model, I should have brought mine in, of the inner ear, take a look at the semicircular canals. It's kind of incredible that the three of them are lined up in the three spatial dimensions, in the XY plane, in the YZ plane, and the XZ plane. They're aligned along the three spatial dimensions, completely orthogonal to one another or at right angles to one another, so that they're able to each sample the three spatial dimensions. At the base of these semicircular canals is this Crista Ocularis. And what this is made of is there's this ampulla. An ampulla, if we look at a semicircular canal, it would be sort of continuing this way, but then would go into this region like this. We call this the ampulla because medicine used to be delivered in what were called ampules. You have the little glass medicine here. This would be scored, and it would snap off, and you administer the medicine. So the structure of the base of the Crista Ocularis looks like an ampule. They call it the ampulla. Inherent in this is this little jelly wedge called the cupula. The cupula has these hair cells with their stereocilia embedded up in that. And as you move, say you do a backflip or something like that, I'm not going to do one right now for you, but the fluid in the semicircular duct is going to begin to rotate along that duct if you're moving that duct in the plane of motion. Right? If you're moving in the plane of motion that semicircular canal has a projection into, is aligned with, the fluid will rotate along that pathway, and it will bend the cupula either this way or that way, depending upon the direction. That then returns information to the brain about this motion of rotation in 3 dimensions. All right. So we talked about the stereocilia. In the hair cells of the Crista Ocularis, is another kind of, it's like a specialized hair cell called the kinocilium. And if the hair cells, the stereocilia, bend away from the kinocilium, it's going to return one kind of information, versus if they're bending towards the kinocilium, it's going to give a different kind of sensory information to the central nervous system. This is going to give you the directionality so that you can tell the difference between doing a front flip versus a back flip, if that makes sense to you. The kinocilium gives a directionality to the information that's returned by the hair cells. All right. So going in one direction causes a depolarization, and going in the other direction causes an inhibition. Now, on to the saccule and utricle. This is where we get our sense of gravity and acceleration. There are the saccule and the utricle are part of this vestibular system. There's two sort of redundant patches in the saccule and the utricle. They are, the primary organ is called the macula, and plural is maculae. There's two maculae, one in the saccule and the other in the utricle. And macula just means patch. It just means a patch. Like there was the macula that the fovea was in the back of the eye. It just means a round patched area. In the macula, there's a bunch of hair cells that are again embedded in this gelatinous material. On top of that gelatinous material is this otolith, ear stone, the otolith, which is made up of these statoconia. Statoconia are little crystals of calcium carbonate. Now, Denise Bruzewitz is my wife, and she has a grandmother who is quite a piece of work. She makes this jelly, jello. She makes this dish called 7-Up salad that she got herself in the McChesney Park calendar with. And 7-Up salad is lime jello made with 7-Up, and on top of it, it has embedded a bunch of bananas and oranges, and then there is whipped cream on top of that, and then sprinkled with, because she's from Wisconsin, shredded cheese. And so this is 7-Up salad, and she claims that it's the family favorite. At least she did in the calendar. And so when I was still dating Denise, she brought me to meet the family, and Grandma brings out this 7-Up salad, and she's bringing it out, and I like in the pit of my stomach, it was starting to like clench up. And she's bringing this jello out, and it's got all this weight. It's a top heavy jello, right? There's all this like stuff on top of it. It's like shaking as she's bringing it to me, right? The jello is kind of like this, and the bananas and the cream and the shredded cheddar cheese or whatever. And nobody else wanted any, but I felt obligated to have some of this stuff. I ate it. And it was burned into my memory, and other parts of me. And it reminds me of this, okay? It reminds me of this because this works in kind of the same way that jello was vibrating. So what happens is when you tilt your head, all this weight from the otoliths on top of the jello kind of slides that jelly down the slope, all right? Slides it down the slope and stimulates those hair cells. It gives you a sense of orientation. It gives you a sense of orientation and gravity. The higher the gravity you are in, the stronger the displacement. And so gravity is nothing but acceleration, right? Gravity is 9.8 meters per second squared. That's acceleration. So if you are in a fast car and you've got the foot to the pedal trying to get out of McChesney Park, that sense of acceleration is also going to work upon the macula of the vestibular system in the ear. All right, you guys following that? Okay, cool. Hey, I made it. Last slide. The vestibular projection pathway. Okay, so all of this information about orientation and acceleration passes along the vestibulocochlear nerve. The vestibular information is going to synapse in the vestibular nuclei in the pons, so that would be, these would be like the first order neurons, and then the vestibular nuclei sends projections where else but the thalamus, so this would be the second order neuron, sensory neuron. And then finally, for us to be aware of whatever information is being conveyed, there has to be a projection from the thalamus to the cortex, and specifically the vestibular cortex, which is a little area that is at the base of the central sulcus in the parietal lobe. This is going to give us our awareness of spatial orientation and movement. There are other projections that come out of the vestibular nuclei that help us coordinate body motion, as you would expect. I don't need to know the names of all those. All right. So I think that's it. Are there any questions?